Hello, today we will learn how to isolate plasmid. So, it is a molecular biology technique which is very useful in normal reactions or setting up different kind of experiments in a molecular biology lab. So, before we start doing this method or of isolating plasmid, we need to know what are the plasmids. So, the plasmid is an extra chromosomal DNA. It usually comes from bacteria, and most of the eubacteria and archaea have plasmids, and these plasmids are double stranded and small in size. So, basically if we look into the size difference of plasmid and the genomic DNA of the bacteria, it differs in the range of few megabase pairs. Like if a plasmid is usually 1 kilobase pair to 1000 kilobase pair, where the genomic DNA of E. coli it's around 4 to 5 megabase pair. So, there is a huge difference in size. Also the plasmid is circular in nature, and it can independently replicate itself. So, why we need to isolate plasmid in molecular biology lab? So, there are different experiments you can design regarding the plasmid isolation, and when you isolate the plasmid, you can design any kind of PCR reaction, you can digest the plasmid, you can clone a particular gene inside the plasmid, also you can do transfection and different other procedures. Now, the isolating a plasmid is a useful technique for microbiology or molecular biology lab, and that is what we are going to do today. So, as the bacteria possess plasmid, they are not essential for their survival, but often this plasmid gives a superpower to the bacteria. So, suppose the bacteria is living in a stress condition, the plasmid can help the bacteria to survive in that particular stress condition, like different antibiotics are present in the media. So, bacteria can survive that kind of condition, because most of the cases plasmid express different kind of antibiotic resistant gene, and this product of these genes are often helpful for the bacterial survival, and we can use this kind of plasmid to easily manipulate or regulate or whatever gene or other product we want from the bacteria, and we can incorporate those gene inside that plasmid and can express that plasmid. Now, the copy number of the plasmid also differs in different strain. There are bacteria which have large copy number of plasmid, or there are small bacteria which have less copy number of the plasmid. During plasmid extraction, you should be careful that which kind of bacterial strain you are using and how many copy numbers are present in that particular plasmid. And also another important aspect of plasmid extraction is that when you are growing the bacteria, the media should contain some kind of selectable marker, like some kind of antibiotic, and that particular antibiotic resistant gene should be present in the plasmid. If you do not put the stress condition, if you do not put the bacteria in a stress condition, the bacteria can eliminate the plasmid. It called plasmid curing. So, the bacteria needs to grow in a stress condition, so that the bacteria will keep the plasmid, and when it will replicate, the plasmid will also replicate itself and make a higher copy number. So, before we start getting into the plasmid extraction, there are few things we need first. One is the bacterial culture. So, usually we give overnight culture. Now, overnight culture means around 12 to 15 hours growth of the bacteria, and this kind of bacterial strain usually there are XL1 strain or BL21 or DH5 alpha strain of E. coli. These have high copy number of plasmid, and they maintain the plasmid in their cell. So, here we are using a LB medium where we have given the bacterial culture. So, LB is an enriched medium. So, you should give an enriched medium where the bacteria can happily grow, and the culture is around 15 hours time. It has taken around 15 hours. So, the OD is usually 3 to 5 at the OD 600. So, you need to have a large number of cells before you start extracting the plasmid from those bacteria. Now, after you have grown overnight a particular culture, then another things which you will need is a centrifuge, because the steps required during the plasmid extraction requires a centrifuge. Also, the solutions which we will need for extracting the plasmid. Now, here today we will use a kit which is provided by a particular company. There are different companies provide this kind of kit. Also, you can make those solutions in the lab following a molecular biology book. So, I will be talking about these solutions and what these solutions contain and how these solutions help us to extract the plasmid. So, three things we need. One is the bacterial culture, an overnight culture, and that bacterial culture should have the antibiotic, which is and the antibiotic resistant gene should be present in that particular plasmid. And another thing is the centrifuge machine, so that we can centrifuge the cells, and also in the later steps we will need the centrifuge and the solutions which will be used. So, now I have mentioned that we need an overnight culture before we go for plasmid extraction. So, here I have given this overnight culture in a culture tube, and if you can see that the culture tube is not fully filled. So, it is around 1/3 or less than 1/3 filled. So, here it is around 7 ml of culture I have given, and I have taken LB and a small inoculum from a cell stock which is kept at -80°C. So, otherwise if you have colony, you can pick one colony and put it inside the culture tube. And also the LB media has a particular selectable marker or the antibiotic resistant antibiotic. In my case, the bacteria contains a PET28 vector. So, the PET28 vector has a kanamycin resistant gene which express when it grows, and so that I have put kanamycin in my media. Other vectors like PET3A have ampicillin marker gene, so you have to put ampicillin. So, depending on the vector or the plasmid you are using, and you have to check what kind of antibiotic resistant gene it contains, and accordingly you have to get that particular antibiotic inside your media. So, it has been grown in the incubator at 37°C because E. coli grows best at 37°C in an incubator. The RPM is almost around 200 RPM. So, the incubator has a parameter where it has to rotate, so that the proper aeration can happen and the bacteria can grow perfectly. So, as I am mentioning, I was mentioning that the culture tube should not be filled too much. So, whenever you are growing bacteria, suppose you are taking any kind of conical or a tube like this, you should not fill that particular container more than 1/3. So, suppose you want to make 250 ml of culture, you should not take a 250 ml or 500 ml conical flask. You should take at least 1 liter of conical flask where it can grow properly. Otherwise, the aeration will be hampered and the bacterial growth will be less. So, now that I have prepared my culture and it has been grown for 15 hours at 37°C, I will isolate first. To isolate the plasmid, I have to take the cell out of this media. So, for that I need to centrifuge this culture in a small Eppendorf tube in multiple go. So, because the Eppendorf tube is almost 1.5 ml and I have almost 7 ml of culture here. Now, today we will use a mini prep kit. So, mini prep means basically you can isolate plasmid from 5 to 7 ml of culture media. If you want to isolate more plasmid, you can isolate up to 50 ml from 50 ml of culture media. You have to use a midi prep kit. And if you want to isolate more like 200 ml or 300 ml of culture media, then you have to use a maxi prep. So, all these different types of kits are available from different kind of brands, and you can take them, or otherwise you can also follow any kind of molecular biology book and you can make those solutions, and accordingly you can step up in your culture volume. So, another thing I would like to mention about here is the one other important instrument which we need during plasmid extraction is the centrifuge. So, centrifuge, this is a simple centrifuge machine where we will isolate the cells from the culture media. So, basically when we will start the centrifuge, it will rotate and it will put a centrifugal force into the different components present inside the culture, and depending on their size and the shape of the molecule, it will precipitate because it has a centrifugal force. So, in every centrifuge there is you have to plug it into any kind of electric channel, and then it has a body switch and you have to switch it on. Now, here the centrifuge has two options, one is the RPM, another one is the RCF. So, RPM means rotational per minute. So, it depends on like how fast it is rotating. This is called the rotor of a centrifuge. So, suppose it is rotating 5000 RPM, so that means per minute it is rotating 5000 times. It does not depend on the size or the radius of the rotor. So, RPM can, so a small centrifuge like this can rotate 5000 rpm, a bigger centrifuge like of different kind can also rotate 5000 rpm, but this rpm does not mean that all the force will be equal to the all the particles that are present inside the solution, because then we come to another term which is RCF, or the rotational centrifugal force, which is applied to the solutes or the different kind of constituents present in a solution. Now that RCF or the G is dependent on the size of the rotor, because then it depends on the radius and also the RPM. So, when you go for any kind of centrifugation, you should know what kind of G force or RCF force you need, because that is kind of universal in terms of centrifugation. Because if someone says that you have to rotate at 5000 rpm, depending on different centrifuge machine and depending on the rotor size, that 5000 rpm can generate different kinds of G force. But if someone says 5000 G, that means in this centrifuge machine, that 5000 G will be similar to another centrifuge machine which has a bigger rotor or a smaller rotor than this. So, you should be careful what kind of G force you are applying for a particular centrifugation procedure. So, here if you see, there is an written as RPM, also you can select for the RCF. So, this is shown as 16162 into G. So, this is the G, and if you go down, this is 14,800 rpm. So, this two value differs from each other, because RPM, basically RCF depends on the rotor size, also the RPM and the gravitational force that is applied into the solution. So, now I will take this culture in a small centrifuge tube and start isolating the cells from this culture. Before that, another important aspect of centrifuge is that you always have to balance the sample. So, suppose you are putting one sample here, at the opposite side you have to put another sample which is of equal weight, otherwise this machine will show error and you will damage the rotor. Another important aspect of centrifuge is that you always have to balance the sample. So, as I am removing the lead of the centrifuge, if I am putting something here, one of my solution, I have to put another sample at the equal at the opposite side of the particular sample, which is of same weight, otherwise it will damage the rotor and it will not give a proper value and the centrifuge will show error. So, you always have to balance your sample. If you have one sample, then you have to balance with some other solution like water or something else, otherwise it will damage the centrifugation rotor. So, now I will take my culture in the small centrifuge tube. Before that, I will label it properly. So, here also I have labeled what kind of plasmid or what kind of culture I have given. It is a good practice, always label your culture tubes or the any kind of culture or any kind of solution you have, label it properly in the lab. So, you give the name of that particular culture which you have given, then you give the date, like when you have put the culture. So, today so I have given the culture the yesterday night, and also you should write your own name, because if you are working in a lab, there are other people and they should also know that you have given the culture. If you do not give a name of your culture or your name, then the problem will be that other people might take your culture, or there might be some kind of mismatch and you might not get the result. So, for easier recognition of your culture, you should give the name of your and the name also of the sample. Also, when you are taking out the culture in a centrifuge tube, you should label that tube as well. So, here I have written the name of my sample in one tube. In another tube, since I am isolating plasmid from only one culture, another good practice in lab is to always label your sample. So, here in the culture tube, I have written the sample name which I have provided or given, and also the date, like when I have put the culture. So, yesterday night I have put the culture, so I have given the sample name, also the date And also I have written my name, because when you are working in a lab there will be other people, and if you do not label your sample properly or write your name, then it might create any problem, like people might take your sample, or there might be some kind of mishandling of the sample. So for that purpose you should always properly label your sample, put down the date and also your name. So always it is a good practice to label the sample properly. It is not only for the culture, in any kind of solution or any kind of buffer you are using, any kind of bottles you have, always label them properly. Otherwise it might create problem, because today you might be remembering what it is, but after 2 days or few days back you will forget what it is, and then it might create confusion of your own. So for that always label your sample properly. Now I will take culture from that sample to another small centrifuge tube. Now those small centrifuge tube also should be labeled properly. So here I have labeled the one centrifuge tube where I will take my culture and pellet down the cells. Since I am isolating plasmid from only one sample, I need balance, as I have mentioned that in a centrifugation procedure you should always balance your sample. So in another tube I have to take water or any kind of sample of equal volume or weight. So I am writing in this tube as balance. So in the balance I will not have any kind of culture, only the water to balance my sample. So now that I have prepared the two tubes beforehand, I will start taking culture from the tube. Now as I have mentioned, this small centrifuge tube is around 1.5 ml. So I have to take multiple times from the tube, because in a mini prep kit you can isolate plasmid from 5 to 7 ml of culture. So as I want to take 5 ml of culture, I will take, using a micropipette I will take 1 ml first. A small volume. Discard the tip. Close the culture tube. Now as I have mentioned, you always need a balance in the centrifuge. So I am taking equal volume of water in the other tube, which is written as balance. And now I will keep the balance, and then at the just directly opposite of the balance I will put the culture tube. Now always remember to put the lid on of the rotor, otherwise it will again show error. Close the centrifuge tube. In the centrifuge there is a mention of RPM as well as the time. So in this centrifuge tube there is no parameter called temperature. Other centrifuge also have temperature probe, but as during plasmid isolation we are not really need the temperature, so it happens in the normal room temperature, which is around 25 degrees centigrade. So we do not need the temperature sensitive centrifuge machine. So it only requires a time and the RPM or RCF. Here we will reduce the time to around 1 minute or 1.5 minutes until there is a pellet formation. And then we will start. Now during this centrifugation what will happen is that there will be a centrifugal force applied to my solution, and as the bacterial cells are quite heavy, they will precipitate down from the culture tube and they will create a pellet at the bottom of the culture tube. So till that time we have to wait till the centrifugation process is over. Now another few things about centrifugation is that if it makes too much noise, or if it is showing error or other things which is not usual in any case, then you should immediately stop the centrifuge machine, because the rotor is rotating at a very high speed. It might cause, if there is any kind of imbalance or any kind of proper leveling was not there, so it might cause some kind of accident in the lab. So you should always be very careful while doing a centrifugation process. So now the timing is going down and it has gone up to 16,000 G. So, how we will know that the pellet down of the cells have been properly done or not? So, when you take out the sample, as I will be showing it to you, the supernatant will be very clear and it will look like the normal media. When you have previously given the media, LV media, it will look like a clear media, and there will be a small platelet formation at the bottom part of the centrifuge tube. Now it is almost over. So, you do not need to stop the centrifuge machine when you give a time like 1 minute or 1.5 minutes. After that timing is over, the centrifuge will itself go down and then it will show me that the centrifugation is over. Now it is over and the lid has been opened. So, now I have taken out the culture tube. As you can see that the supernatant is highly clear and there is a small pellet formation at the bottom part of the centrifuge tube. So, this pellet means these are the cell pellets from the E. coli bacteria and the media has been cleared. So, during this kind of experiment, you should always have a small beaker where you can discard all this flow through. So, any kind of liquid discard, you should not throw it in a normal dustbin. You should always take a clean beaker, because this kind of solution have media, and if you throw it in a normal basin or wash basin, it can contaminate the environment. So, you should take it in a clean beaker, and then it needs some kind of treatment, like we put detergent, also some bleach, so that other microorganism like other bacteria and fungus cannot grow further. So, now I have taken around 1.2 ml. I will again take the same culture in the same tube, as I have discarded the media. So, the tube is now free. Again in the same tube I will take more culture. Now, as I have discarded the supernatant from this culture tube, you can see only the pellet is there and the tube is free now. So, I will be taking another 1 ml of culture in the same tube, so that around 5 ml of culture all the cells are taken into a same tube. So, I am taking almost 1.2 ml. You do not need to be very precise about this, because anyway we are going to take all the cultures present in this cell. So, I am not changing the balance, because I took almost same amount as previously. Always remember to put on the lid, close the rotor and again start, and always keep an eye. If the machine is making any kind of noise or showing errors, immediately stop the centrifuge machine. So, now again we have to wait for another 1 minute, and in the same culture pellet there will be another 1 ml. So, another 1 ml all the cells will be into a pellet. So, my second round of centrifugation is also over. So, it will stop on its own. So, now you can see the RPM value is going down, that means it has been stopped, and it will gradually stop and the lid will open on its own. Again you can see the upper part of the supernatant is almost clear, as if like a normal media, and there is a big cell pellet formation at the bottom of the centrifuge tube. Again I will discard the supernatant into this beaker. Do not throw it here and there. as the tube is free now or empty, I will take another 1 ml of culture. Again put it in the at the opposite side of the balance, run it for another time. If you are changing the volume too much, then you need to change the balance volume as well. Otherwise, if you are taking every time a similar volume, then you do not need to change the balance volume every time. If it shows any kind of error or the balancing is not proper, then you should change the balance volume as well. So, here I am actually pelleting down the cells at the highest speed for 1 minute or more than 1 minute 15 seconds. If you feel that the supernatant is not properly clear or the cell pellets are not properly formed, then you can increase the RPM or RCF, or you can increase the timing as well. So, in most cases if you are pelleting down from 50 ml or 15 ml of culture, we go for 10 to 15 minutes around 3000 or 5000 G. If you do not have that much, at high speed 1 minute is enough. So, you should always check the supernatant. If you feel like the supernatant is not properly clear, then you go for higher speed and or higher time. My third round is also done. Again the supernatant is clear. I will again discard the flow through. And again I will take another 1 ml. Put it at the opposite side. If you feel like you do not know exactly which one is the opposite side, you should count the blank holes in the rotor. So, from here there is 1, 2, 3, 4 and 5. Again from this side it is 1, 2, 3, 4 and 5. So, that means it is equally distant. So, it is at the exactly opposite side. Now, as I was mentioning that the copy number varies in case of different strain of E. coli. So, basically if you feel when you isolate the plasmid, if you feel like the plasmid isolation or the content of the plasmid is very poor, the yield is poor, then it might happen that the bacterial strain whichever you are using has low copy number. Otherwise, if you feel like, so in those cases if you have lower yield, you can, which problem hochhe? Okay, আমি আরেকবার থেকে শুরু করছি. So, as I was mentioning, the copy number varies in different bacterial strain. So, if you feel like that yield of the plasmid extraction is low, then you can go for a higher volume. Suppose here I have given 7 ml of culture, then you can give 10 or 15 ml culture to get higher yield. You should check the copy number of the bacteria as well, like how many plasmids are present in one bacterial strain. So, in normal cases the BH5 alpha have around 10 to 15 or hundreds, even hundreds or more than hundreds as well. So, if the bacterial strain contains low copy number, then the yield will be less. So, for those cases you need to go for higher culture. If the bacterial copy number is very high, then 5 ml to 7 ml is fine. Also, it will depend what kind of subsequent work you will do with the plasmid. If you want to do PCR, very less amount of DNA is okay with your experiment. If you want to do restriction digestion, any kind of ligation or transfection, depending on your necessity, it will change. Now, my last round of centrifugation is also done. So, now again I will throw away the culture or the supernatant. Here I have taken a piece of tissue paper. I will just dab it I will invert the tube and wrap it so that all the media has been soaked away and the pellet is as much as possible is without the media. So, now we will start the purification of the plasmid. So, there are different types of purification method. In this case, we will be doing alkaline lysis method. So, as I have mentioned earlier that I will be using a mini prep kit, but you can make this kind of solution in your lab as well, following any kind of molecular biology protocol book. So, the steps which we have to follow will be given by the prep kit manufacturer. Also, it is a similar protocol, you can get it online or follow any kind of book. So, there are 3 types of solution. Which we will need. The first solution is so today we will be using a mini prep kit, and the steps which we have to follow will be given by the kit itself, like it will be there inside the kit, or you can follow any kind of internet protocol or any kind of book. The protocol will be similar in most of the cases. So, there are few solutions which we will need. The first solution will be the resolubilizing buffer. So, as I have pelleted down my cells, I need to again take these cells into a buffer. So, there are few things we need, few solutions. The first solution which we will need is the resuspension or resolubilizing buffer. So, as I have pelleted down my cells, I need to take these cells into a buffer solution where other solution will work into. So, the resuspension buffer is kept mostly at the 2 to 8 degrees centigrade. So, that is why I keep it in a ice box. So, this buffer contains trace, a buffering agent. Also, it has EDTA which will chelate all the divalent ions like magnesium and manganese. So, these divalent ions are necessary for the action of the different kind of nuclease. So, when subsequently we will break down the cells, the nucleases will be out of the cells and it might damage the DNA which we want to extract. So, for that the EDTA is there in the resuspension buffer. It also contains RNAs. Since we are isolating DNA and we want to take only the DNA part, there might be some contaminant from the RNA part. So, we degrade those RNA using an RNase. So, this resuspension buffer contains a buffering agent which is trace, EDTA which will chelate down the divalent ions, and RNase A which will kill the RNA. After resuspending this buffer, then we need to break down the cell walls or the cells. So, for that we need to have a buffer which is the lysis buffer. So, lysis buffer is mostly alkaline, that is why the name comes alkaline lysis method. So, it has high amount of NaOH or sodium hydroxide. Now, sodium hydroxide will actually denature all the proteins that are present. It will denature the membrane proteins, anything which are protein related will be degraded. Also, it has anionic detergent which is sodium dodecyl sulfate. So, sodium dodecyl sulfate will actually break down the cell membrane and it will help to lyse the cell. So, this lysis buffer contains high amount of sodium hydroxide. So, it is an alkaline solution. It will also denature the DNA or RNA whichever is present, whatever is present inside the cell. So, everything will be denatured while we were giving the lysis buffer. Now, after giving the lysis buffer, the timing is important. So, in the kit it is written around 4 to 5 minutes is enough, more than 5 minutes should be avoided, because we want the denaturing process is very time sensitive. So, do not keep the sample in denaturing buffer or the lysis buffer more than 5 minutes. After this we need to have a neutralizing buffer. So, this buffer is mostly 3 molar of potassium acetate. So, potassium acetate is acidic in nature. When we will give in this buffer into the lysis buffer, what it will do is it will readily The third buffer which we will need is the neutralization buffer. So, neutralization buffer has around 3 molar of potassium acetate. So, this is acidic in nature. Now, immediately after giving the potassium acetate buffer or the neutralization buffer, it will start the reannealing of the DNA. Since the previous buffer or the lysis buffer has strong alkaline substances, it will denature all the protein as well as the DNA and RNA. After you put the soluble neutralization buffer, it will start reannealing the DNA. So, as I have mentioned earlier, the size differs in case of plasmid DNA and the genomic DNA. Where the plasmid DNA is from 1 kb to mostly 1000 kilobase pair, the genomic DNA is from 4 to 6 megabase pair. So, immediately after you give the neutralization buffer, the plasmid DNA, since it is very small and also it is very covalently close, the two strands are very close to each other, it will readily anneal together. So, you will get the plasmid DNA in the solution, whereas the genomic DNA will require much more time to properly anneal. The two strand will be haphazardly annealed and it will form a clump and it will precipitate out of the solution. Also, as I have mentioned, the potassium acetate is there in the neutralization buffer, it will form potassium dodecyl sulfate. So, the potassium dodecyl sulfate is a salt which is again unsoluble in the solution, so it will precipitate out, and along with that precipitation all the proteins and other contaminants that are present in the solution will also precipitate out. So, when we will put the solubilization neutralization buffer, we will see small curd like substances forming, and those contains the proteins, other contaminants and also the genomic DNA. And immediately after putting the neutralization buffer, we will centrifuge the whole solution, so that other substances will precipitate out and the plasmid DNA which is covalently linked with each other and they can readily anneal after we give the potassium acetate, and it will be kept in the solution and we will isolate the solution. Now, after taking out the solution which will contain the plasmid DNA, and in this kit we will use a column. So, column is basically a silica membrane. So, when we will give the solution inside this, at the bottom part of this column there is a silica membrane and our plasmid DNA will get stuck to the silica membrane. And then what we have to do is we need to wash the column to get rid of all the other